Cytoplasmic organelles. Organelles are the metabolically active and permanent residents of the cell. Organelles exist in two forms, membrane bound and not bound by membrane. The principal cytoplasmic organelles include mitochondrion, ribosome, Golgi bodies, endoplasmic reticulum, chloroplast, lysosomes, peroxisome, nucleus. Mitochondrion. Unlike most cytoplasmic organelles, mitochondria are large enough to be seen in the light microscope, and their presence within cells has been known over 100 years. As with most other organelles, mitochondria possess recognizable morphological characteristics while exhibiting considerable variability in appearance. The typical mitochondrion is sausage shaped, approximately 0.2 to 1 micrometer in cross sectional diameter and 1 to 4 micrometer in length. In some cells, such as those of early embryos, mitochondria are almost spherical in shape, whereas in others, such as fibroblasts, they are elongate, thread-like structures. An average mammalian liver cell contains about 1500 mitochondria, corresponding to 15 to 20% of the cell's volume. Mitochondria are even more numerous in muscle cells, which require large amounts of ATP to fuel their contraction. A particular striking arrangement of mitochondria occurs in sperm cells, where they are located in the cell's mid-piece just behind the nucleus. The movements of the sperm are powered by ATP produced in these mitochondria. Mitochondria are also prominent in many plant cells, where they are the primary suppliers of ATP in non-photosynthetic tissues, as well as a source of ATP in photosynthetic leaf cells during periods of darkness. The internal structure of a mitochondria is as shown in the image. Each mitochondrion contains an outer membrane and a complex inner membrane system. The outer membrane completely encloses the mitochondrion, serving as its outer boundary. Although part of the inner membrane may lie just inside the outer membrane, much of it occurs as deep folds or invaginations called cristae. In some cells, as in those of the mammalian liver, the cristae are wide sheets that across the entire diameter of the mitochondrion. In most plant cells, the cristae have a more tubular shape. The infoldings of the inner membrane greatly increase the amount of surface available to house the machinery needed for aerobic respiration. The membranes of the mitochondrion divide the organelle into two aqueous compartments, one within the center of the mitochondrion called the matrix, and a second between the outer and inner membrane called the intermembrane space. The matrix has a gel-like consistency owing to the presence of a high concentration of water-soluble proteins. Mitochondrial membranes. The outer and inner membranes have very different properties. The outer membrane is composed of approximately 50% lipid by weight and contains a curious mixture of enzymes involved in such diverse activities as the oxidation of epinephrine, the degradation of tryptophan, and the elongation of fatty acids. In contrast, the inner membrane has a very high protein to lipid ratio, more than 3:1 by weight, which corresponds to approximately 1 protein molecule for every 15 phospholipids. At least 60 different polypeptides are found within the complex structure of this membrane. The inner membrane is virtually devoid of cholesterol and rich in an unusual phospholipid, cardiolipin. Both features are characteristic of bacterial plasma membranes, from which the inner mitochondrial membrane is presumably derived by evolution. The outer mitochondrial membrane is thought to be derived from an outer membrane found as part of the cell wall of certain bacterial cells. Both contain porins, integral proteins that form large non-selective membrane channels. Because of the porins, the outer membrane is especially permeable, allowing molecules up to about 10,000 Daltons to pass freely into the intermembrane space. The inner membrane is highly impermeable. Virtually all molecules and ions require special transporters situated in the inner membrane to gain entrance to the matrix. Mitochondrial matrix. In addition to various enzymes, the mitochondrial matrix also contains ribosomes. Mitochondria possess their own genetic material and the machinery to manufacture their own RNA and proteins. This non-chromosomal DNA is important because it encodes a small number of mitochondrial polypeptides that are integrated into the inner mitochondrial membrane together with polypeptides encoded by genes residing within nucleus and imported from their site of synthesis in the cytosol. Functions of mitochondria. Mitochondria play a critical role in the generation of metabolic energy in eukaryotic cells. They are responsible for most of the useful energy derived from the breakdown of carbohydrates and fatty acids, which is converted to ATP by the process of oxidative phosphorylation. Most mitochondrial proteins are translated on free cytosolic ribosomes and imported into the organelle by specific targeting signals. In addition, mitochondria are unique among the cytoplasmic organelles already discussed in that they contain their own DNA which encodes tRNAs, ribosomal RNAs and some mitochondrial proteins. The assembly of mitochondria thus involves proteins encoded by their own genomes and translated within the organelle as well as proteins encoded by the nuclear genome and imported from the cytosol. The matrix contains the mitochondrial genetic system as well as the enzymes responsible for the central reactions of oxidative metabolism as shown in the image. The oxidative breakdown of glucose and fatty acids is the principal source of metabolic energy in animal cells. The initial stages of glucose metabolism or glycolysis occur in the cytosol where glucose is converted to pyruvate. Pyruvate is then transported into mitochondria where its complete oxidation to carbon dioxide yields the bulk of usable energy or ATP obtained from glucose metabolism. This involves the initial oxidation of pyruvate to acetyl-CoA, which is then broken down to carbon dioxide via the citric acid cycle. The oxidation of fatty acids also yields acetyl-CoA, which is similarly metabolized by the citric acid cycle in mitochondria. The enzymes of the citric acid cycle, which are located in the matrix of mitochondria, are central players in the oxidative breakdown of both carbohydrates and fatty acids. The oxidation of acetyl-CoA to carbon dioxide is coupled to the reduction of NAD+ and FAD to NADH and FADH2 respectively. Most of the energy derived from oxidative metabolism is then produced by the process of oxidative phosphorylation, which takes place in the inner mitochondrial membrane. The high energy electrons from NADH and FADH2 are transferred through a series of carriers in the membrane to molecular oxygen. The energy derived from these electron transfer reactions is converted to potential energy stored in a proton gradient across the membrane, which is then used to drive ATP synthesis. The inner mitochondrial membrane thus represents the principal site of ATP generation, and this critical role is reflected in its structure. First, its surface area is substantially increased by its folding into cristae. In addition, the inner mitochondrial membrane contains an unusually high percentage of proteins which are involved in oxidative phosphorylation as well as in the transport of metabolites between the cytosol and mitochondria. Otherwise, the inner membrane is impermeable to most ions and small molecules, a property critical to maintaining the proton gradient that drives oxidative phosphorylation. In contrast to the inner membrane, the outer mitochondrial membrane is freely permeable to small molecules. This is because it contains proteins called porins, which form channels that allow the free diffusion of molecules smaller than about 6,000 Daltons. The composition of the intermembrane space is therefore similar to the cytosol with respect to ions and small molecules. Consequently, the inner mitochondrial membrane is the functional barrier to the passage of small molecules between the cytosol and the matrix and maintains the proton gradient that drives oxidative phosphorylation.